Okay, so we have some time for questions. First of all, I just wanted to make one comment. Obviously, I think early on, Ramon and Stephen both talked about how important our mission is. It's really around targeting, helping patients with cancer. And we have, two physician scientists on the panel who actually see these patients every day. And actually, one reason we highlighted them is that they have the largest and most developed programs, really ranging from the standard clinical care all the way to these translational research programs. And I know many of you are not clinicians, but we have many other programs here that we have not been able to highlight. And one of the things that the Mount Sinai Health System has done is really created one of the largest cancer programs in New York and really in the country. We have as many new cancer cases in the Mount Sinai Health System as Memorial Sloan Kettering. And this is a huge opportunity, and as we recruit and grow these programs, everything we want to do is really ultimately to be translational for our patients. So I want to remind you of that and all of the opportunities that we have as you develop these targets and want to work with clinicians. We have to create a way to connect you. So the first question I have is for you, Poulikos, which is, what's the mechanism of action of SHP2 inhibitors? Are there other proteins that can be targeted, and what about melanoma?
So the mechanism of action is a little different than, so there's some history to the development of inhibitors for SHP2. Initially, they were just catalytic inhibitors, but they couldn't distinguish between SHP2 and SHP1, which is important actually SHP1 is necessary, so these were extremely toxic. So more lately, they do, I think it's Novartis mostly, but there's another company as well. They took advantage of an allosteric pocket. So these inhibitors lock SHP2 in an inactive conformation, and they disrupt the complex that forms with the receptor, GRIP2 and SOS. And of course, I think that even in melanoma, it's the same basically idea. The Nature paper that was on vemurafenib, they actually plugged in monitoring on phospho-ERK 2 weeks after giving vemurafenib. And there was a clear correlation between response and inhibition of signaling. So I don't think that even in melanoma, the end of the road has been reached in terms of how well the pathway is inhibited. But I think in colorectal and thyroid, because it's a huge unmet need, colorectal BRAF V600E, even though it's 10% of colorectal, it's actually a larger population than the 50% of melanomas that have V600E, and there's nothing there for these patients in terms of targeted therapies, of course.
Thank you. Yoko, so people are excited about the possibility that the PDX model is working, but the question is, can this be done in real time for a patient as they come in?
The engraftment process does take several months, up to for most of our models. What we hope to do is to be able to create a collection that's broad enough that captures the heterogeneity of most of the triple-negative breast cancer subtypes. And because we're deriving primary cultures and cell lines from these PDX mouse models, we hope to also supplement the database with characterizations, not only the genomics, but sensitivities to inhibitors from, for example, FDA-approved libraries, to have that information. And then when a patient shows up in clinic and has their tumor biopsied clinically and profiled on an RNA, DNA level, can we match that to an existing model that's already out there, annotated with clinical information along with genomics and drug sensitivities, such that we could maybe propose that this particular patient's tumor may respond to this experimental drug in the context of a clinical trial. So there are many ways to think about it. I think we're also developing metastatic models, which in many cases is very time sensitive, as Ross has previously described, but we are attempting to utilize these models in multiple different ways such that we can benefit patients as a group as well, and not as well as an individual.
Because you still have only 1/3 of them growing, so do you think we'll ever technically get to a level where you can grow it in every patient, the PDX?
Yeah, so this is one of the first studies where primary tumors are being actively sought and engrafted. Most of the literature involves engrafting metastatic lesions because those have a higher engraftment rate. Our engraftment rate is only about 30% right now, but we are looking at different models right now to supplement some of these mice so that the engraftment rate is higher. Like IL-6.
Well, I was actually wondering about organoids or other systems that would also complement this and maybe faster. We looked into that.
So the primary cultures and organoids derived from the models would be tested against a panel of in screens, not only of inhibitors, novel PROTAC agents, but also the beauty is we have the mouse model to see as an intermediate step maybe that the drugs might work.
So somebody wanted to ask, this was to Joel, but maybe you can answer it, Samir. Do network models work better than doctors at predicting what therapy patients should receive?
So I just want to emphasize that all the work we've done is actually meant as a decision support system for doctors, not as a replacement.
Okay, thank you. I still have kids going to college, so, okay.
To pay off our medical school debt.
All right. So one question for you, Gian, is a specific one around this ALK PROTAC. Is it an ALK1 or pan-ALK? Does it work for ALK2? And I guess that gets at maybe the development, just maybe you could also expand it if when you have a target, basically how broad do you decide to create a drug that will cover multiple structural versions of your target?
That's a great question. So the PROTAC technology is fairly new, and so it's in early days, and large rules are being defined. But for example, of course, ideally you would like to start with a compound highly selective, inhibitor or binder, highly selective or specific to the protein target. Okay, right? But in the case such binders is not available, right? So then you could start potentially a compounds binds to several close related targets, but selected for those targets over other targets. But it has been demonstrated in these cases, selectivity can be achieved even though the binder part of it binds to several protein targets, but selective degradation can be achieved to a single target. Okay, so that has been demonstrated. But in terms of actually I would say the advantages of the PROTAC technology or the PROTACs over traditional inhibitors or binders, one potential big advantage is, let's see, you have really well-known oncogenes or the targets, and there's no effective small molecule inhibitors.
Right.
But then you somehow actually have binders to those proteins, but they are weak binders, binds at allosteric binding site, and so therefore do not have functional activity. Okay, so now you can actually convert that binder, which is not active functionally, into PROTACs, which would eliminate the entire oncoprotein. Okay, so that's one advantage. So another potential advantage is in terms of drug resistance. So why the PROTAC can do that? Because let's see, the resistance, the tumors develop resistance, and then the mutations that impact the binding of the inhibitors to the drug target. So the binding affinity is significantly reduced. However, for PROTAC to work, and the binding affinity does not require it to be really high, so therefore you can convert a weak binder and then convert that into an effective PROTAC, therefore can be potential approach for drug-resistant tumors.
Great. Very exciting. Thank you to everyone in the session. It was really perfect.